So we'll move on to informatics and data. Nick's already referred to Rosalind and to David, and again, you all know David. David Hume, Director of Rosalind Institute and Research Director at the Edinburgh Vet School. David.
Thanks. I guess I reciprocate Kim's viewpoint. Of course, I perhaps feel a little insecure being up here as a straightforward academic. I can, with all honesty, tick the box when I go back into Australia that says, have you been on a farm recently, and say, no. So we tend to operate at a different level in the supply chain, and I think the exciting thing about working with CL is actually joining up the different sectors of the supply chain, giving us a path towards a two-way innovation. I think a lot of applied scientists that I've worked with in the past are well-balanced individuals. They have chips on both shoulders. That's a good line. That they think that the basic scientists look down on the applied science, and in fact the reality is that it's a two-way thing, that the problems come back and there is a genuine virtuous circle. This is one of the slides that we actually showed when we presented the concept of CL to the nice people at Innovate UK, where we looked at the network of inputs and interactions between CL and the innovation pipeline, and CL sitting in the middle. And we realised that we would bring a whole lot of expertise into a central hub, if you like, of knowledge and data. And that would include expertise and knowledge coming from our international partners. We have just recently established with SRUC a partnership with the International Livestock Research Institute in Nairobi. And that's actually very interesting because they are interested in production traits, but they have the resilience traits. And that's where we might look for animals that are more resilient in our environments, bringing some of the partnerships back. We obviously have the industry partners. We have the other centres, including those funded by Agritech. We have the commercial farms. We have a network of veterinary service providers and vet schools across the whole of the UK, an unprecedented collection, really, if you think about I think 4 vet schools, 3 major research active veterinary service providers covering all of the sectors. We start to get information back from processors. We have experimental farms, and we also have our production scale experimental facilities across all of the sectors, some of those actually running different species side by side and starting to think about how you can integrate production systems with more than one species on a single farm. And of course we bring information back from the retailers and ultimately the consumers. So I don't have terribly sophisticated slides, but I'm going to talk briefly about informatics and what we intend. There is actually the very small amount of recurrent funding from the CL is put towards hiring staff to create a livestock informatics hub. That hub in Edinburgh will involve ourselves and SRUC and will also involve the Bill and Melinda Gates funded ILRI Centre because we have exactly the same parallel infrastructure in that centre. So we'll start to get a critical mass of people who can deal with the data deluge, if you like, that we've already heard a little bit about. When we come down to what we're trying to find out, this is the simple the genomics view, if you like, of how you end up with an animal. You have a particular genotype, you have an environment, and you end up with a phenotype. And if you dissect that out, you have a particular genotype of animal plus its microbiome, and those two things interact with each other. We know that there is genetic control of the microbiome, so it's not just an adventitious addition to our species. Humans have their own microbiome, which they lovingly share with animals and backwards and forwards. So We work, for example, on Staph aureus that does nice host jumps into other species, comes back to humans again in an altered form, and zoonoses as well are also part of this microbiome. We have the animal health products that maintain, if you like, some kind of barrier, including, of course, the antibiotics. We have the form of nutrition, we have the production systems, and we end up with livestock products and hopefully improved productivity. Now, most of these inputs have actually been optimized separately. And so if we look at the genetic input, particularly into the highly stratified industries like poultry and pigs, we have the breeding companies that are taking independent male and female lines, optimizing the female line for female relevant traits and the male line for production traits like growth and meat. Then you end up with chooks that are a four-way cross, or pigs that are a three-way or four-way terminal cross. And the breeding companies are very good at optimizing the animals at the top of the breeding pyramid, and they know nothing at all about the performance of the animals at the bottom of the breeding pyramid, nor is there any really serious comparative data on the performance of the products from different suppliers in different production systems. And frankly, they don't want it. There's no breeding companies here today, so I can say that. Of course, we want to work with them. What we also have started to realise biologically is, and there's a rather spectacular piece of science recently from my viewpoint, is that there is much greater contribution of different phenotypes from the male and female lines in a cross. And in most breeding pyramids, especially in ruminants, we genotype only the male line, and we infer the optimal bull from the performance of his female progeny and ignore the genetic contribution of the females. And just to give you a little bit of science, a recent paper showed that around half of the genes that are looked at, only the male or only the female partner's contribution is actually expressed in at least one tissue. So that means it makes a difference whether you inherit the gene from the male or the female line, and we don't even look at that currently. So that's a bit of a diversion into science. So we don't breed animals for optimal use of animal health products. It's quite obvious that we could breed animals that respond better to vaccines. We could breed animals that have a better ability to convert certain feed inputs. We could breed animals rather obviously for performance in different production systems. For example, we haven't seriously bred chickens to modify pecking behaviour for many years, and the result is then you put them in these lovely enriched cages. I'm sure some of you have seen them. At the end of a laying season, there is one fully fledged bird in the cage, and all of the others are in various stages of being pecked to death. That I've seen this in action, so it's obvious that we could also, in these open pig production systems, the pigs are not social animals when they're breeding. There is aggressive behaviour. We know that there's a genetic control, and so we could breed pigs for behavioural traits. So what it comes down to, in order to start to address these questions, is we need better genotyping and we need better phenotyping. And the genotyping side of things is what we do, and it's getting disturbingly cheap. It's the cost of genome sequencing, which I'll come down to, is coming down drastically. It's becoming feasible to create genotyping platforms for an individual population of animals. So we're currently working on a project with the Irish Cattle Breeding Federation to genotype every cow in Ireland, and that is now feasible. It's also feasible to take your pedigree Jersey herd and actually genotype every animal in it and optimise the crosses within your pedigree herd, and it's now cost effective. And so with the genotyping revolution, we need, I think what CL is about, genotyping is becoming trivial, is actually collecting phenotypes, novel phenotypes, better phenotypes, comparative phenotypes, and I think that, for me, coming from my side of things, that's the exciting part of what we can do. I said, I mentioned genotyping. So Edinburgh Genomics in Edinburgh was formed from ARC Genomics, which was the major livestock sequencing facility in the UK funded by BBSRC, and we merged with the medics and the environmentalists into a single facility. We're fortunate to have on the doorstep the UK supercomputer, which has massive storage capacity, so we can actually provide the infrastructure to CL for a major informatics hub, and also for very cost-effective whole genome resequencing. So it costs about £800 now to sequence a cow at terminal depth, and that price is still coming down. Genotyping platforms, I think we're going to come down to breed-specific platforms that will be £5 to £10 a throw very soon, which becomes cost-effective. Microbiome sequencing, we no longer do ribosomal RNA. If those of you who know that area, we simply sequence the whole lot, and that has become more cost-effective and far more informative. And then, to me, one of the exciting things is rapid diagnosis of pathogen outbreaks. So instead of trying to isolate the pathogen, we simply sequence the lesion, and we can assemble the pathogen genome by just subtracting the mammalian sequence. So it's giving you a taste, and I think the way that can be applied across this network of CL, this may sound like a Roslin-centric proposition. This is an asset that I think is actually going to be democratised in a sense over the next 5 years, and it will be no longer something that Roslin offers with a great competitive advantage. These new sequencing machines will be this big, and they will put out sequence in real time. So the other side of things is phenotype, and what is actually being invested in at the Edinburgh site is a large animal research and imaging facility. This is the intensive end, if you like, of the research offering of CL. We are in the business of genome editing, which I think is going to revolutionise livestock. There's obviously a policy issue involved there, but it's becoming disturbingly easy. To do that, we need experimental surgeries. We have an intensive care unit for large animals, which is partly used for medical research, but enables us to look very intensively at the physiology and biology of production animals with a multi-scan CT at the level of a human hospital and 3 Tesla MRI. We produce, as Nick mentioned, pathogen-free chickens, and we're looking for funds for a large pathogen-free pig production facility to provide animals of very highly defined health status for experimental facilities across the UK. So I won't belabour that, but we also have environment-controlled facilities where we'll be able to look at infectious disease challenge with very high intensity monitoring of the animals under controlled conditions. So that's the major applications of that for industry partners, obviously in the transgenics and genetics, in development of animal health products, developing new reproductive technologies, which is one of the things that we do a lot of in the avian space, as mentioned by a couple of people, developing technologies for better assessment of meat and carcass quality, and also increasingly developing new approaches to understanding brains and behaviour. Another one of my favourite pieces of science at the moment is what's called optogenetics, which actually involves putting a light-sensitive neurotransmitter receptor in part of the brain and then just shining a light on the animal and changing its behaviour. I'm not sure that's going to happen in the future, but I could imagine a time when we have transgenic chickens tunable, and we're collaborating with green gauge lighting to get exactly the right light for the right chook. In fact, Greengage Lighting is one of our partners. This facility is actually, thankfully, with a fairly complex and rapid negotiation, is going to be co-located with the northern hub of the Precision Agriculture Centre, which I think gives us really lots of opportunities to join up those two centres. So that's me.
Great.